Today, we'll be doing a mind map on the adaptations of photosynthesis. This is a continuation of my mind map on photosynthesis, where I discuss the mechanisms, reactions, and processes involved in photosynthesis. You can go and check that out. However, today, we'll be looking at the features of the plant, particularly the leaves, which aid the efficiency of photosynthesis. First, let's look at leaves. When you're young, you're probably taught that plants grow towards sunlight. That is true, and this can be seen in leaves, where they change their arrangement to face the sun and prevent being blocked by other leaves. This is to ensure that there is maximum absorption of sunlight. How leaves do this is through phototropism, which is discussed in depth in my mind map on plant science and structure. The leaf also has a rich network of veins which is made up of xylem and phloem tissues. This helps photosynthesis as the xylem provides a constant supply of water to leaf cells. This is essential for the reactions in photosynthesis and other features of the leaf, as we will see. The phloem, on the other hand, transport the glucose that is produced by photosynthesis away from the leaves to the rest of the plant. Glucose is transported as sucrose in the phloem. The petiole, or stalk of the leaf, holds the lamina upright and away from the stem of the plant. This allows the leaf to obtain the maximum amount of sunlight and air, both of which are essential for photosynthesis. The lamina is often spread out and has a large surface area. This ensures that the leaf absorbs the maximum amount of sunlight. The lamina is also very thin. This allows carbon dioxide in air to rapidly reach the inner cells of the leaf for photosynthesis to occur. To better understand the adaptations of the lamina, we will have to look at its cross-section. There are 6 main features of the cross-section of a leaf that you need to take note of. The first is the cuticle, which is a waxy layer on the surface of the leaf. It reduces water loss from the plant through evaporation. The next layer of cells are epidermal cells, referred to as the upper epidermis. Water is constantly supplied to the epidermal cells so that the cells maintain its turgidity. This ensures that the leaf is as spread out as possible to maximize its surface area and hence absorption of sunlight. The same goes for the lower epidermal layer. The Palisade mesophyll layer is packed tightly with Palisade cells arranged in a vertical manner. The reason for this arrangement is to maximize the exposure of Palisade cells to sunlight for photosynthesis. The bulk of photosynthesis occurs at the Palisade mesophyll layer, where it also has more chloroplasts compared to the spongy cells. This is because the Palisade cells are positioned closer to the leaf surface, where more light energy can be harnessed. The spongy mesophyll layer is just below the Palisade mesophyll layer, where spongy cells can be easily identified by its irregular shape. The irregular spongy cells create numerous large intercellular air spaces. This allows for the rapid diffusion of gases in and out of the leaf. This includes carbon dioxide, which has to be taken in for photosynthesis, and oxygen, which is released as a product of photosynthesis. To assist this diffusion in and out of the mesophyll cells, they are covered by a thin film of moisture. This allows carbon dioxide to dissolve in it and diffuse into the cells quicker, and oxygen to diffuse out as well, which escapes via the stoma. Stomata are openings in the epidermal layer that allows for gaseous exchange. This includes the diffusion of carbon dioxide, oxygen, as well as loss of water vapor from the leaves. They are usually located at the lower epidermal layer. Guard cells surround each stoma, which regulates the opening and closing of the stoma. In the presence of sunlight, stomata open and become bigger. This increases the intake of carbon dioxide for photosynthesis, hence increasing the rate of photosynthesis when sunlight is available. How guard cells control the stomata is through osmosis. In the presence of sunlight, 2 things happen. Firstly, as the guard cells contain chlorophyll, they photosynthesize in the presence of light to produce glucose. Secondly, potassium ions get pumped into the guard cells from its neighboring epidermal cells. Due to this, the water potential in guard cells is lowered, leading to the movement of water from epidermal cells into the guard cells via osmosis. The movement of water into guard cells causes them to become turgid, hence increasing the size of the stoma. The reason why guard cells form stomata is because the cell wall on the side where the pair of guard cells face each other is thicker. Therefore, when the cell becomes turgid and expands, the cell does not expand equally, leading to an opening being formed. Therefore, this is how the rate of photosynthesis increases, as a larger stoma means that there can be a larger intake of carbon dioxide. On top of increasing the rate of photosynthesis, having a bigger stoma also increases the rate of transpiration. Transpiration is the loss of water vapor from plants, primarily through the stomata on leaves. Transpiration occurs so as to draw water and mineral salts out from the roots and distribute it throughout the plant. Transpiration is able to do this because when water is lost from the plants through the leaves, it creates a difference in water potential between the leaves and the roots. The difference in water potential creates a suction force in xylem vessels, referred to as transpiration pull. Transpiration pull is transmitted throughout the xylem, which draws water from the soil into the roots of the plant and pulls it up to the leaves. This ensures a constant supply of water and mineral salts to the plant, particularly the leaves. This is advantageous for photosynthesis as water is an ingredient in photosynthesis. Therefore, a constant supply of water will help maintain a high rate of photosynthesis. A constant supply of water to the leaves also helps keep epidermal cells turgid. This ensures that the leaves remain spread out for maximum absorption of sunlight. The loss of water vapor through the stomata is also necessary to keep the leaves and the plants cool, similar to sweating. This prevents leaves from being scorched or damaged. There are 4 factors that can affect the rate of transpiration. The first factor is sunlight, which affects the size of stomata, as we just discussed. When stomata on the leaves are bigger, water is more easily lost, hence increasing the rate of transpiration. The next factor is temperature, where higher temperatures increases the rate of evaporation of water from the plant. The plant also loses more water so as to keep itself cool and prevent scorching. Wind speeds affect transpiration as high wind speeds blow water vapor around the stomata away, which creates a water vapor concentration gradient between the intercellular air spaces in the leaf and the air outside the stomata. Higher wind speeds create a steeper gradient, hence increasing the rate of diffusion of water vapor out of leaves via stomata. The effect of air humidity has a similar explanation, where the amount of water present in air changes the water vapor concentration gradient. A higher humidity means that there is more water present in air, which lowers the concentration gradient, and vice versa. How water is lost is through the evaporation of water from the film of moisture around cells in the intercellular space. Water evaporates to become water vapor in the intercellular space, where it subsequently exits the leaf via the open stomata. The 4 factors I just described affects the rate of transpiration by either increasing the rate of evaporation of water from the film of moisture or increasing the rate of movement of water vapor out of the leaves. Water can also evaporate from the laminar surface. However, the cuticle tends to prevent this. Water that is lost is replenished by the xylem, which provides a constant supply of water to all cells in the leaf. The xylem is part of the vascular bundle, where vascular tissues act as the transport system in plants. The vascular bundle also consists of phloem vessels, which transports food in the plant. Glucose, produced by photosynthesis, are loaded into the phloem as sucrose, which get transported to other non-photosynthesizing parts of the plant. If not used by the plant, it can be stored as starch. We will discuss more about these 2 vessels in my mind map on plant science and structure. I've also put a link in the description to a 3-dimensional cross-section of a leaf, so that you can better visualize how the vascular bundle works and looks like, because a 2-dimensional visualization just does not serve justice. As you can see, the xylem and phloem are separated by a layer of cells called the cambium. Cambium tissue contains undifferentiated meristem cells, which is essential for secondary growth. You will be able to understand this better by looking at a cross-section of a stem. As a plant grows, xylem and phloem tissues develop as the stem grows thicker. The cambium is crucial for this growth, as the undifferentiated meristem cells differentiate to form more xylem and phloem tissues. Do note that the cambium is a continuous ring in stem and not necessarily just in between the xylem and phloem vessels. The xylem is usually on the inside of the stem, while the phloem is on the outside. However, you can easily identify the xylem by its large open vessels, as xylem vessels are made up of hollowed out cells. I try to show this with my illustration on the left. However, I recommend you Google a proper cross-sectional photo to better understand what I mean. Once again, I'll talk more about this in my mind map on plant science and structure, so I highly recommend you check that out as the topics are related. Before we wrap up this mind map, let's take some time to look at how water is taken in by the plant. As we are able to see by now, water is very important in photosynthesis, not only as an ingredient in the reaction, but in its role as part of the multiple adaptations of photosynthesis. So I think it is only fair that we understand how water gets into the plant in the first place. Water is absorbed from the soil into the plants at the roots. A cross-section of a root can be seen here. The outermost layer is the epidermis, which is made up of epidermal cells. The epidermis is what is in contact with the soil and is where water first enters the roots. Some epidermal cells are modified to have tubular outgrowths, referred to as root hairs. The epidermal cells with these tubular outgrowths are called root hair cells. At the very center of the root is the vascular bundle, where the xylem and phloem are situated as such. The vascular bundle is enclosed by a ring of cells called the endodermis. The space between the endodermis and the epidermis is the cortex. Therefore, once absorbed, water has to travel from the epidermis to the xylem vessels to be transported to the rest of the plant. This is how water moves from the soil into the xylem vessels. Water coating the soil particles are absorbed by epidermal cells via osmosis and aquaporins, which are channels in the membrane of the cell. To aid this absorption, vacuoles in epidermal cells are filled with a concentrated solution of sugars and salts. This helps establish a low water potential in the cell compared to the surrounding soil particles, which facilitates the movement of water into the epidermis via osmosis. The root hairs of the epidermis also help the absorption of substances into the plant by increasing the surface area to volume ratio of the roots. After being absorbed at the epidermis, water moves through the cortex to reach the endodermis. Water moves through the cortex by either an apoplast or symplast pathway. The apoplastic pathway is when water moves from cell to cell through the cell wall in the space just outside the plasma membrane. The symplastic pathway is when water moves from cell to cell through the cytoplasm in the space just inside of the plasma membrane. Plasmodesmata, which connects cells, allows for this direct flow between them. After passing through the cortex, water reaches the endodermis, which acts as a screening layer. The cells at the endodermis layer screen the substances before they enter the vascular bundle. Therefore, its role is preventing harmful substances that may have been absorbed from entering the transport system of the plant. Finally, water enters the xylem vessels, where transpiration pool will force water up the vessel to the rest of the plant. This is how water is absorbed from the soil into the plant. Roots do not only take in water, but other substances such as mineral salts, often in the form of ions. These solutes are taken in at the epidermis by various methods. Solutes can firstly enter the root via diffusion through the plasma membrane. Solutes may also enter via mass flow, where solutes are dissolved in water and flows into the plant together with water. Lastly, solutes can be taken in via active transport, which usually occurs by either co-transporter membrane proteins or voltage-gated ion channels controlled by energy stored in a proton gradient. On top of having tubular outgrowths of root hairs to increase the surface area for absorption, another adaptation that roots have are the Hyphae network that branches from the roots. Hyphae is a branching filament of fungi that grows on the roots. The network of Hyphae has a large surface area in contact with the soil, which channels water and nutrients into the roots, hence increasing the rate of absorption. This is an example of mutualism, where there is a mutually beneficial relationship between the fungus and the plant. The fungus aids plant absorption of water and other nutrients through the Hyphae network, and in exchange, the plant supplies the products of photosynthesis to fuel the metabolism of the fungus. This brings me to the end of my mind map on the adaptations of photosynthesis. Do check out my other mind maps on plants, as the topics are all linked. Anyways, I hope you find today's mind map useful and have a good day.